Okay, if you are in Wednesday or Tuesday lab, please make the observation on the board that Dr. Morgan put up there about what you're supposed to do with your plates. Quiz Monday will be on DNA replication. That's sections 13.2 and 13.3 in the chapter we're working on right now. So please be ready for that. Do you have any questions? Okay, let's see if you were paying attention yesterday, Tuesday. I obviously wasn't. You've got a chance to get 2 bonus points if you're paying attention. I put something on the board on Tuesday that was just wrong. Anybody catch me on it? If you did, quickly jot it down on a piece of paper and pass it forward. I would like to say that I was testing you to see if you were paying attention, but I just had a brain freeze. Let me ask it this way. Is anybody going to attempt and can answer? Okay. What did it do? It was in the context of talking about the cell cycle. And the cell cycle is divided up into various phases. The M phase, the G1 phase, the S phase, and the G2 phase. What did I put on the board? I had S down here, prior to the division. So you go through this G1 or kind of preparation for DNA replication, and then after DNA replication, there's some other things that happen within the cell, and then it goes through the M phase, which is the mitotic phase of the cell cycle. Okay? If you ever see me making a mistake like that, and you are aware of it, please call me on it, okay? I know there are many times when students will go, that's not right, but you won't say anything, okay? We're not infallible. We make mistakes too. Okay, let's continue our story of DNA replication and the structure of DNA discovery. So here's our major players, Watson and Crick, looking at this model that they have developed. The purpose of building a model is to then test experimental evidence to see if it is consistent with the model. A model is really a hypothesis. Okay? So this was their hypothesis of the structure of DNA. And then they had to start looking at available information about DNA and say, is this particular model consistent with the information? Because if it isn't consistent, what do you have to do to the model? You have to modify it, okay? Your hypothesis has to fit your data. We talked a little bit about some of the information they already had that they could test their model against. And one of the things that they could test their model against was this concept of Chargaff's rules. Remember what Chargaff's rule said? We had to do with base pairing, and basically it said that there is this equivalency in DNA, no matter what source it comes from, where the amount of A equals T and the amount of G equals C. There was other information that was available about DNA. That was obtained through a technique called X-ray diffraction. And X-ray diffraction studies of DNA were being carried on at a very close by college to Cambridge, where Watson and Crick were, called King's College. And this is where Maurice Wilkins and Rosalind Franklin were working on investigating DNA through this X-ray diffraction analysis. Now, X-ray diffraction is a technically difficult technique. It takes a lot of expertise to interpret the results. And as it turns out, it was a dangerous technique as well. Because as they were using X-rays, they were being exposed to X-rays. At that particular point in time, we weren't quite as aware of how damaging X-rays can be. And Rosalind Franklin died quite early in life before she could be recognized with a Nobel Prize, they think because of cancer resulting from all kinds of exposure to X-rays. What happens in X-ray diffraction? Well, you've got some kind of a source that generates X-rays, and that X-rays that are produced are focused and shined on a crystal of a molecule, like DNA. And as the X-rays pass through this crystal, the atoms in the molecule start diffracting or bending the X-rays so that they are coming out of the crystal at all kinds of different angles. And the angle that they come out of the crystal is dependent upon the structure of the molecule that's in that crystal. And then they've got a photographic plate that can be exposed by those X-rays coming through the molecule, and that generates a pattern. So it's not a picture of the molecule, but it is really a representation of how this molecule is scattering or diffracting these X-rays that are passing through it. And here is one of the more famous photographs that was obtained by Rosalind Franklin of X-ray diffraction of DNA. And this really doesn't tell you much to the untrained eye, but Franklin knew how to interpret these particular photos. And one of the things that this X-ray diffraction pattern suggested to her was that the molecule was a helix, a spiral molecule. And there was also some physical information about the DNA molecule that you could obtain by X-ray diffraction. And don't worry about these numbers, but I want you to understand kind of some of the physical parameters that she was able to deduce about the molecule from that photo that she had obtained. And there's an interesting story about this photo. James Watson kind of got this photo without her permission and used the insights from this to come up with their model of DNA replication, and then basically chose not to credit her for her contributions. So Watson was kind of a scientific creep in that respect. So what did this X-ray diffraction data tell you about DNA? It suggested that it was actually a double helix, and so there were two strands of DNA wrapped around one another, and that this double helix had a uniform width. Okay? So if you look at this molecule right here, that the width of the molecule was consistent from one end to another. It wasn't wider in some places and narrower in other places. And that actually turned out to be quite important in figuring out the final structure of DNA. The nucleotides are of two types. Adenine and guanine are the purine nucleotides. Cytosine and thymine are the pyrimidine nucleotides. And from this information, they were able to determine the distance between those nucleotides in the DNA double helix, and it was about 0.34 nanometers. They were able to determine that there was a regularity with which this helix twisted, and they knew the physical parameters of that, so the helix made a turn every 3.4 nanometers, and that there were 10 layers of nitrogen bases for every turn of the helix. Well, that makes sense, because if these are stacked 0.34 nanometers apart, 10 of those would give that 3.4 nanometer single twist to the DNA. So this was crucial information for Watson and Crick. So they just kept making models, and they kept testing their models against the available evidence to see if the model made sense. Initially, James Watson really believed that in the double helix, that like nucleotides paired with like nucleotides. And so he believed that adenine always paired with adenine, thymine always paired with thymine, etc. But when they started to test this idea against the model and the evidence, it really didn't make sense. Purines are bigger than pyrimidines. Okay? Purines have this 2 heterocyclic ring structure. Pyrimidines only have a single heterocyclic ring structure. And so if like paired with like, some regions of the DNA would be pushed farther apart, and other regions of the DNA would be closer together. And so the DNA molecule would be kind of lumpy. X-ray diffraction data suggested that the diameter of the DNA molecule was consistent. It was always the same. And the only way you could do that was to get a purine paired with a pyrimidine, a big one with a little one. So adenine and thymine. Adenine's the purine, thymine's the pyrimidine. Guanine and cytosine. Guanine's the purine, cytosine's the pyrimidine. Well, the paper that they published really was the birth of molecular biology and genetic engineering, which is what we're going to be doing in lab for the rest of the semester. And as I mentioned before, it was a single-page paper. That is unheard of now, particularly one that had such a fundamental effect on all of science. In this same journal of Nature, Wilkins and Franklin actually published their X-ray diffraction data as kind of an accompaniment to this particular paper. There is a copy on the website. I would like you to look at it. It's only a page, but I think it's important to kind of look at this seminal paper that was the birth of molecular biology. And what were the major insights that were revealed in this particular paper? Well, DNA was a double helix, that the 2 DNA strands are held together by hydrogen bonds between complementary base pairs. Adenine pairing with thymine, guanine pairing with cytosine. And finally, DNA molecules are anti-parallel. And that's important, and that's going to become very important when we talk about replication of DNA. So, here is a chemical representation of a single strand of DNA. Remember, DNA is actually 2 of these strands twisted around one another. But here is a single strand, okay? Here's a nucleotide, here's another nucleotide, here's another one, here's another one, okay? Remember, each nucleotide consists of a phosphate group attached to a sugar, and then attached to one of 4 of these nitrogen-containing bases, adenine, thymine, guanine, or cytosine. This is a polymer, or a chain. The monomers are these nucleotides. Within a given DNA strand, any nucleotide can be attached to any other nucleotide. Okay? How does the attachment occur? The phosphate group of one nucleotide forms a covalent bond with the sugar of the adjacent nucleotide. So we form a sugar-phosphate bond. That particular bond is called a pyrophosphate bond. So basically, in this single strand of DNA, this forms kind of the backbone of the molecule, and then hanging off of each of these sugars are one of 4 nitrogen-containing bases. In this strand, they could all be A's, or they could all be T's. There's any possible number of different combinations that these could be. Okay? Another thing that you need to really pay attention to is this strand of DNA has polarity. There is a distinct head end, if you like, and a distinct tail end. The head end is referred to as the 5' end. And that is the end that terminates in a phosphate group. Why do we call it the 5' end? Because the phosphate is attached to the number 5 carbon on the sugar. That's where that 5' designation comes from. At the opposite end of the strand, the tail end, if you like, the molecule terminates in a sugar. Okay? That's designated as the 3' end. Okay? So 5' end, phosphate, 3' end, sugar. If I add another nucleotide down here, it's still going to terminate in a 3' sugar molecule down at the bottom. Okay? The base pairing that occurs between nucleotides in the 2 strands of DNA is said to be complementary. The base pairing consists of adenine on one strand, hydrogen bonding with thymine on an adjacent strand. There are 2 hydrogen bonds that stabilize the interaction of these 2 nucleotides. So on this model of DNA, right here, this white rubber part would represent the sugar phosphate backbone. And then protruding off of that would be these nitrogen-containing bases. Okay? So, here are 2 nucleotides that are complementary to one another, guanine and cytosine. They are held together by a hydrogen bond. Okay? The bonds that join the sugar and phosphate together are stronger. These are covalent bonds. Okay? So these are going to be hard to break. But these hydrogen bonds that bond the adjacent nucleotides together on the 2 strands are relatively weak hydrogen bonds. Okay? Guanine and cytosine, when they're bonded together, form 3 hydrogen bonds. Adenine and thymine, 2. So these are a little bit stronger than adenine and thymine base pairs. Why is it important that the DNA double helix is held together by hydrogen bonds as opposed to covalent bonds? What would be the problem if these were covalent bonds?
They wouldn't be able to be separated.
They wouldn't be able to be separated. It would be hard to break those bonds. And one of the things we know is that during replication is that this strand temporarily has to separate from this strand. And both of these strands will serve as templates, we'll find that term in a minute, for making a new strand. Okay? But the fact that these have only hydrogen bonds, doesn't that make DNA kind of unstable? And will it have a tendency to kind of fall apart? Potentially. But a lot of little weak bonds collectively can make the molecule relatively stable. Okay? What's something that could cause this DNA molecule to start falling apart and having those hydrogen bonds broken? Heat energy, a change in pH. All of those could affect the interaction of those hydrogen bonds, and those could cause the DNA molecule to denature. Okay? So that's one of the reasons why in the cell, we really need to maintain the internal temperature of the cell at a regulated temperature. And if the cell gets too hot, molecules start denaturing. Proteins start unfolding, DNA molecules start separating into their 2 strands, and that's not good for the cell. But when the cell is ready to replicate, these bonds do need to be broken so the 2 template strands can be separated so replication can occur. So, this shows you the 2 strands with the complementary base pairing that occurs between those strands. Shows you some of the physical parameters of the DNA molecule that were determined by X-ray diffraction. So the radius of the DNA molecule here the distance between one twist in the molecule here, distance between adjacent bases here. But again, please note, you can have any order of bases on one of the strands, but what's on one strand dictates what's on the other strand because of the base pairing rules. Okay? So I can have a G here, an A here, a T here. This could easily have been another A or C. So once I've got this G over here, you've got to have a C across from it. So basically the sequence of one strand dictates the sequence of the complementary or partner strand. This is just a space-filling model showing you some of the atoms associated with the DNA double helix. This blue-yellow region right here is that sugar phosphate backbone right here. And then these regions in here represent the complementary base pairs that are sticking inward. In one of the early models, Watson and Crick thought that the base pairs were on the outside and that the sugar phosphate backbone was actually on the inside. But that was consistent with the evidence that they had. This race to discover the structure of DNA was a very hotly contested race. And I think I mentioned on Tuesday that in addition to the group in England working on this particular question, there was a world-famous biochemist, Linus Pauling, at Caltech, who was also working on this. And the smart money was on Pauling because he already had a Nobel Prize. He was a world-known biochemist who had done a lot of work with the three-dimensional structure of proteins. Pauling came up with a model of DNA replication that was a triple helix, and he was convinced he was right. And he was ready to publish. And his nephew happened to know some of the people at Cambridge, and Watson and Crick got a look at a preprint of the proposed publication. And they said, we're done, we're toast, Linus has figured this out. And they opened it up and looked at this model of a triple helix, and they started looking at it and they went, he's wrong, this can't make sense. Because our X-ray diffraction data, which he didn't have access to, is just inconsistent with his particular model. So they continued on, and they ended up being right. So Linus Pauling didn't always get it right, even though he was a Nobel scientist. Remember that I said that one of the things that stimulated an interest in discovering the structure of DNA was that hopefully once you understood structure, you could understand questions of function. So for example, how does DNA replicate? How does DNA encode genetic information? Watson and Crick, once they figured out that this was the structure of DNA, had a very aha moment. They looked at one another and said, I know how this replicates. And as a matter of fact, at the very end of their one-page paper, they had one little sentence at the very end that said something to the effect that it has not escaped our notice that this model of DNA structure immediately suggests an explanation for how DNA replicates. And they believe that DNA replication occurred in a fashion that is now referred to as semi-conservative replication. Here's the idea. Very simple conceptually. Here's a little stretch of DNA that we would want to replicate. Okay? Here's one strand. Here's the complementary strand. Sometimes people will refer to these as the Watson strand and the Crick strand. Okay? But again, notice the complementary base pairing. If we can separate these two strands from one another, then each of those can serve as a template for making a new strand. The idea of a template actually comes from industry. Template means a copying surface. And if you can make a copying surface, then you can make new copies of that from that surface molecule. So if these two replicate and we start to make one new strand that's a copy of this and one new strand that's a copy of this, This information will determine what the sequence of the newly copied strand is. So, let's use this designation. This is our parental DNA molecule, okay? And then during replication, the parental molecule separates. The strands unwind, okay? So that's this one, that's this one. Now, we need to use each of these as a template to make a new strand. And we will illustrate the new strand as a dotted line, so you represent that as being a newly made strand. So these would represent the two daughter DNA molecules. Okay? So if this is a chromosome, then this would be the duplicated chromosomes. Okay? So this strand would serve as a template for making this strand. This strand would serve as a template for making this strand. And if you look at it, if you got an A here, the new strand's got to have a T. C, G, A, T. So this is one daughter molecule, this is the second one, and their sequences are both identical to the original parental molecule. Make sense? Okay. Very simple, very elegant. So this is the semi-conservative model. The reason that we call this semi-conservative is that in the daughter molecules, one of the original parental strands is saved or retained in the daughter. Okay? So, according to Watson and Crick's belief, if this is our DNA double helix right here, replication would begin at one end of the molecule as the two parental strands separated, lighter color here. And then each parental strand would serve as a template for making a complementary strand, so that this would be a daughter DNA molecule, this would be a daughter DNA molecule. Well, what did I say Tuesday about ideas in biology? Just because something is logical, does that necessarily mean it's true? No, we found that in photosynthesis. Other people who started studying DNA replication said, okay, it makes sense that replication would be semi-conservative. But we can envision some other possibilities by which DNA might replicate. There are dispersive mechanisms, there are conservative mechanisms that potentially could be in play. What would those look like? Okay, so here's our semi-conservative. Okay? Parental strands are indicated in dark blue. Newly made strands indicated in light blue. Okay? So after one round of DNA replication, if replication is semi-conservative, each daughter strand would consist of one parental and one new. Okay? After a second round of replication, this particular strand right here would make a new daughter strand. This light daughter strand would make another light daughter strand. This. So you get a little bit different pattern of what the DNA molecules look like after the second round of replication versus the first one. Okay, so these wouldn't have conserved the parent. In dispersive, here's our original DNA molecule, and according to dispersive, each daughter strand is a mixture of old and new. And then each subsequent daughter strand would generate a mixture of old and new. So this would generate a different pattern of kinds of molecules than this one did. In conservative, what they suggested here is that the original DNA molecule is entirely retained, and the other daughter strand is entirely newly made DNA. So then this would form entirely new DNA. So would this. But this molecule would be still entirely parental. Now, it was really hard for people to envision how these forms of replication could occur. This one made perfect sense, okay? Like this. But you can't discount these just because you can't understand how they might work. So, in 1958, two researchers, Messelson and Stahl, devised an experiment to try to answer this question. What is the method of DNA replication? Is it semi-conservative? Is it conservative? Or is it dispersive? They used two tools when they devised their experiment. They used isotopes of nitrogen and they used centrifugation. The most common isotope of nitrogen is nitrogen 14. So its mass is 14. However, there are other isotopes of nitrogen in nature, including N15. If N14 has 7 protons, atomic number 7, and therefore 7 electrons, how many protons does N15 have? That's 7, right? There's still nitrogen. But what's this bottom, this number down here? That's a mass number. What's mass consist of? Protons and neutrons. So N15 has 1 additional neutron. So which is heavier? N15 is heavier, right? It's heavy nitrogen. Okay? And what does centrifugation separate molecules on the basis of? Density, or weight, or heaviness. Okay? So if you could prepare DNA where you could label it with N14 and N15 nitrogen and then follow that through the process of replication and separate the resulting molecules by centrifugation, you could figure out which of these particular patterns was correct. So what they did was they grew bacteria in liquid culture media that contained N15 labeled proteins, which they used as their source of nitrogen. They grew the bacteria in there for several generations. So basically all of their DNA that had been replicated now contained this heavy nitrogen. And then they took some of this bacteria out and transferred into culture media, which contained the lighter nitrogen. So this is what they were going to use to make their new DNA molecules, that lighter nitrogen. They let these bacteria grow in this lighter nitrogen media for 20 minutes. What's significant about 20 minutes? That's the approximate time it takes for DNA molecules to replicate once. They did another experiment where they let these bacteria in the media for 40 minutes, and during that time, DNA had replicated twice. So go back to this diagram we had here for a second. So the dark blue would represent heavy DNA. Okay? The light blue would represent light DNA. This is the nitrogen 14 that these cells are now growing on. So, if DNA replication is semi-conservative, after the first 20 minutes, if you look at the DNA, both of the daughter molecules should be a mixture of heavy and light. After 40 minutes, 2 of them should be a mixture of heavy and light. 2 of them should be entirely light. What if it is conservative? After 20 minutes, 1 molecule should be heavy, 1 light. After 40 minutes, 3 of them will be light, 1 will be heavy. With dispersive, it's all mixed up here. They're all mixtures. They're all mixtures. So we can actually determine which of these outcomes occurred in this experiment by centrifuging the DNA after replication and separating on the basis of weight. So which of these would go farther down in the tube? Which would pellet at the bottom? The heavy DNA. Which would be in the middle of the tube? The hybrids, the ones that had a mixture of one strand light, one strand heavy. And what would be at the top? The ones that were entirely light. Okay? So they did the experiment. And for 20 minutes, one round of replication, these were their predictions. Okay, here you would have one light band, one heavy band. Here you would have mixtures of light and heavy. When they did the experiment, they found a mixture of light and heavy. So that refuted the argument of conservative replication. But it still left open the possibility for either semi-conservative or dispersive. Okay, what if you did the experiment for 40 minutes or 2 rounds of replication? Okay. Well, here's what you would expect after 40 minutes in conservative. They didn't see that. Here's what you would expect after 40 minutes of dispersive replication. One band. You didn't see that. They did see 2 bands. One of these intermediate bands, N14, N15, and one of these light bands. So this was strong evidence in support of the semi-conservative model of replication. The model that made the most sense appears to be the one that the cell uses. But I think it is important to understand that we can't just dismiss these other possibilities just because we can't see how they might work. Okay? So in this particular case, the logical one did seem to be the one that the cell was really doing. Well, as it turns out, semi-conservative DNA replication is not really as straightforward as this Watson and Crick early concept had suggested. That the molecule starts unwinding at one end, templates separate, and we keep making 2 new strands until we reach the opposite end. Okay. Here's some things that kind of contribute to DNA replication being more complex than Watson and Crick originally thought. So let's look at some of these considerations about DNA replication. Okay, if DNA is going to replicate, it's got to be able to unwind. How does it do that? Unwinding involves breaking hydrogen bonds. Why do those hydrogen bonds break only prior to replication? Why do they not break at other times? DNA replication is fast. Okay? Excuse me. In bacterial cells, new DNA can be made at a rate of about 500 nucleotides per second. Okay, so 1001. A stretch of DNA, 500 nucleotides has been copied. That's blazing. Okay? It's a little bit slower in mammals. We think about 50 nucleotides per second. Mammalian chromosomes are more complicated than prokaryotic DNA is. Okay? It's incredibly accurate. When you look at replicated DNA, on the average, there is a mistake in the base pairing of about 1 nucleotide for every 1 billion replicated. Now think about this for a second. Okay? If you were given something on a sheet of paper and said, I want you to type an exact copy of that on your computer, you're probably going to make a mistake while you're typing, right? And to have an error rate of only 1 mistake for every billion letters is incredibly good. Here's something that's really important. Okay? There's an enzyme that is very much involved in copying DNA. It's called DNA polymerase. And DNA polymerase has a couple of limitations. One limitation is it can't start replication of a new strand by itself. there has to already be something there for that polymerase to attach a nucleotide onto. Once there are nucleotides in place, DNA polymerase can begin to add new ones onto existing ones. But how does this first nucleotide get there? DNA polymerase can't do that. Another limitation of DNA polymerase is it only works in one direction. DNA polymerase will only work to build new strands in their 5' to 3' direction. We'll talk about that more in a second. Finally, remember that DNA molecules are anti-parallel. So one is head to tail, the other is tail to head. And when you resynthesize or copy DNA, you've got to maintain that polarity. Okay? So, if this is the 5' end, and this is the 3' end, and this is 3' and 5'. So these original strands are anti-parallel. Okay? When we copy, this parental strand has to retain that polarity. So does this one. So the idea that DNA replication can occur by just unwinding the molecule at one end and then making this strand over here and this new strand over here doesn't fly. Because if this one is being made in the 5' to 3' direction, this one is being made in the 3' to 5' direction. And the enzyme can't do that. It can't make a new strand in the 3' to 5' direction. That's a problem. That's something that Watson and Crick had no idea about. Well, how does DNA address and solve some of these concerns or considerations? Well, the process of DNA replication involves several proteins. And most of those proteins are enzymes. So we need to spend a little bit of time talking about some of the replication enzymes and what their roles are. Okay, so what was the first consideration? The DNA's got to unwind prior to replication. How can we do that? Well, there actually is an enzyme called DNA helicase which breaks the hydrogen bonds between the two DNA strands and allows them to separate. This happens at a site on the DNA molecule called the origin of replication. If we take this DNA molecule and start unwinding it and start breaking these hydrogen bonds and untwisting this double helix, something interesting happens at this end of the DNA molecule. The molecule becomes all twisted up. So as we unwind it here, and these are still intact, this region just gets all tangled up. And that's going to interfere with replication later on. When it's all tangled up, it can't unwind. Well, there's an enzyme called topoisomerase. And what it does is as the DNA is replicating at one place, it acts on the other end and makes a little nicks in the DNA molecule and allows those twists to kind of unkink. And then it seals them back up again. So it keeps on twisting one end as the other end is unwinding. Got to have that or DNA replication won't occur. When these two strands separate, what's the tendency going to be for those two strands to do? We've got a double helix held together by hydrogen bonds. We break the hydrogen bonds and the two strands separate. What's the natural tendency of that molecule going to be to do? come back together again, right? For those hydrogen bonds to reestablish. Well, there are some proteins that associate with the DNA as it untwists called single-stranded binding proteins. And they temporarily associate with the DNA and keep that double helix separate long enough so that the replication can occur. It prevents it from coming back together before it's had a chance to replicate. So that is 3 of the proteins involved dealing with just this one consideration. How can replication be so fast? Well, one of the things we know about enzymes is that enzymes are able to increase the rate of chemical reactions. And the major copying enzyme that is used in replicating DNA, DNA polymerase, really speeds up the rate of DNA replication. In bacteria, there are 3 major forms of DNA polymerase. And the major replicating one is DNA polymerase 3. In our cells, we've identified to date about 11 different types of DNA polymerases. And we probably haven't figured out the role that all of those play in replication yet. But there's this enzyme basically that grabs nucleotides, A's, G's, C's, and T's, and hooks them onto this newly growing strand of DNA. And which nucleotide is hooked on is dictated by what's the nucleotide in the template. So this is the main replicating enzyme. This is the one that causes the polymer to be formed, the DNA polymerase. Another thing about DNA replication is that in DNA, replication doesn't typically occur from just one end of the molecule to the other. In DNA replication, it actually ends up occurring in 2 directions simultaneously. So, here's a strand of DNA. And according to Watson and Crick's belief, replication would start at one end and just proceed all the way to the other end. In reality, we think that there are all kinds of little origins of replication that are initiated simultaneously on the same DNA molecule, and they call this a replication bubble. And that these bubbles then proceed in both directions away from one end to the other. And as this bubble begins to merge with this bubble, begins to merge with this bubble, then the entire DNA molecule is copied. This is going to be faster than just forming one bubble and having it go from one end of the molecule to another. And in this electron micrograph, you can actually see some of the replication bubbles that are being formed as DNA is being copied. So this will help to speed up the rate of DNA replication. Okay, what about accuracy? How can we get that 1 billion mistake rate, 1 out of a billion mistake rate? Well, the DNA polymerase enzyme actually has proofreading capabilities. So as it brings in a new nucleotide when it's copying DNA, after it puts it in place, it almost like it looks at it and says, okay, does that belong? And if it doesn't, it takes it out and puts the right one in. It's almost like autocorrect on your word processor. Okay, you will occasionally hit the wrong key and a word will be misspelled. But the software will autocorrect that and say, I know what you really meant. This is the word. Okay? So DNA polymerase has that ability to proofread itself and autocorrect, which really minimizes the number of mistakes that can be made. Okay, I think I'll leave this one for next time because we only have a couple minutes, but the big one is some of these limitations of DNA polymerase. How can we make a DNA new strand if DNA polymerase can't start it on its own? How can we make new DNA molecules if the replication enzyme only works in one direction, and the DNA molecules are anti-parallel. We'll pick up with that tomorrow.
Okay, well, it's pretty
Just any time before the next exam. Sure.
What was the name of the movie you were supposed to ask? You're just good on there and just...
Oh, did you get the email I sent?
Yeah, I
It's a YouTube link,
okay.
so you don't have to get it from that link.
All right.